and welcome to the National Association of Plant Breeders webinar series, Building Better Plants for the 21st Century, Updates and Upgrades in HTP, IP, and DNA. So my name is Keisha Chen, and I'll be serving as your host for the webinar. Today's presentation will be given by Dr. Kevin Price from AgPixel and the Department of Agronomy at Kansas State University. Dr. Price researches biogeography with specialization in remote sensing, geographic information systems, and spatial analysis. The emphasis of his research is to elucidate the application of drone remote sensing technologies in agriculture. Today, Dr. Price will be speaking about the use of technology for high-throughput plant phenotyping in his presentation. How can ultra-high resolution drone imagery be acquired and used for high-throughput plant phenotyping? So now I'm going to switch control of the screen over to our speaker, Dr. Price.
Well, thank you for joining us, everyone. As you saw, my name is Dr. Kevin Price. I'm the Executive Vice President of a company called AgPixel. We also have a part of our company is called RoboFlight Systems. We work with drones and Cessna and satellite imagery. The company, just a couple of slides about us, and we'll get on to the technical issues and so on. We have about 40 years of remote sensing experience among our team, 35 years in commercial aviation, and 30 years in unmanned aircraft system operations. A little background, I was a professor for nearly 30 years. I taught at Utah State University, and you see here I taught at the University of Kansas, and I taught at Kansas State University. I was in the Department of Agronomy and started working with drone imagery for research in agriculture in about 2011. A little more background. I've served as an advisor on drone technology to the National Research Council in agriculture, the Committee in Agriculture and Natural Resources, as an advisor to a former U.S. Secretary of State, and to NASA and to NOAA, and have testified before congressional and Senate subcommittees. So that's enough on that. Just want to let you know a little bit of my credentials. In terms of drone technology, I'm going to start off talking about drones in general, but I want to make a point. Remember, the drone is the platform. It's not the end product. Here you're seeing different types of drones. There's helicopters. I put this picture of the Predator up here because Kurt DeMuth, by the way, can you see me moving my mouse? Mark, are you able to?
Yes, we can see your mouse, yeah.
All right, so I'm circling around our COO, Kurt DeMuth, who was trailing or escorting the Predator back into the United States. Whenever it comes into U.S. territory, it has to have an escort. Here you're looking at, I'm working on different types of drones, and I'll explain the different types in just a second, and the advantages and disadvantages. What makes a drone is that a drone, a small one that weighs under 55 pounds, is really a model airplane with autonomous guidance system. So the basic difference between it and a model airplane is it does have autonomous flight capability. There's two types of drones. There's what we call fixed wing, as you see on your left, and then there's multi-rotor, as you see on your right. For most high-throughput phenotyping work, you're going to see me suggesting that this type of an aircraft, the multi-rotor, be used. The fixed wing in a single day, flying on a good day at under 400 feet, which is what the FAA allows, you can map about mappable quality imagery about 1,000 acres. These are channel numbers. With a multi-rotor, you're looking at 50 to 100 acres if you're doing map quality, which means you're looking straight down and getting images that are not highly oblique views. And I've said here, here's a close-up of the multi-rotor. One of the advantages of the multi-rotor is you can usually carry more weight. And you see a camera here attached to a gimbal on this multi-rotor, which helps you keep the camera pointing straight down. Whether it's pointing straight down or not is not absolutely critical, because the software design nowadays will allow you to factor out some of the oblique viewing angles that you might get. Here's a video showing us doing some high-throughput phenotyping work on winter wheat at Kansas State University. A couple of my graduate students were flying this. You see the green and orange pads that to tell us which way the aircraft is oriented, so we're flying in the right direction. You can fly these low and slow, which lets you get a lot of really good detail. Now the agronomist or the plant breeder is embarrassed when I show this because the plots are not all lined up nicely. There was a problem obviously with the GPS that day or something. So we got a drone above a drone, so we're getting the video. So these are pretty easy to fly. The question is how good of photography you get on. This is what we call color infrared photography with some calibration tools out. So the redder it is, the cleaner it is the way you should interpret this. You can see we got enough detail. We can literally, when at a different stage, when there were seed heads on the winter wheat, there were seed heads. Yes.
We really can't do the sound is so loud really hear you very clearly, so you may want to either pause and talk and then let the video play, or because the sound of the video is sort of drowning your voice out.
Thank you.
Sure.
All right, here's an example of a fixed wing. This is actually one of ours we build for our dealers. We're not really in the market of selling drones, so please don't interpret this as an attempt to sell a drone. That's not the purpose here. But with the right training, this is an agronomist from Christina from Canada, up in Canada. She was flying canola. She flew that aircraft for 56 minutes. Got 1-inch resolution imagery, and each of the black dots on here is an image position or a photograph position. To give you an idea, there were 1,424 images captured during that 56-minute overflight. On the left here, you're seeing the part of our portal where we make all the data accessible to the people who need it. Now, this is a fixed-wing aircraft I'm flying. One thing you're going to see in here is 4 different bays for putting cameras. If you're going to do aerial imagery, you want an aircraft that will allow you to put cameras of multiple types, maybe a thermal scanner or several different types of cameras. Okay, that gives you an idea of the 2 different types. The last one you saw is much more difficult to fly for a person who's not used to flying aircraft, especially model airplane type of aircraft. But if you know what you're doing, they can be a very effective tool as well. Okay, I'm going to shift now from talking about the drones, we'll come back to them a little more, into talking about some basic remote sensing. I have no idea what your remote sensing backgrounds might be. What I'm going to do is talk about color infrared photography and how it's used and how we use it to compute indices. We're basically looking at energy within the electromagnetic spectrum. The visible portion of the spectrum we perceive with our eyes, but then if you move into the longer wavelengths past the red, which is the longest wavelength we can detect, you get into what's called the near infrared, and then if you want to go into thermal, you go way out far into the short wave, clear out in this area as opposed to over here in the visible or the near infrared region. What we're talking about when we talk about plants is the detection of chlorophyll density. We're looking at wavelengths that are sensitive to chlorophyll absorption. And so you can see here a graph showing that the chlorophyll absorbs wavelengths in the blue region and over in the red and yellow region of the spectrum. And the index that I'm going to show you is sensitive to the density of chloroplasts per unit area, which is the description of density, obviously. Here I've got a plant, a soybean plant on a table in a greenhouse, taking a picture of it. The side that I'm circling now is the healthier part of the plant. Over here on the left side, the plant is more diseased and affected. It's more chlorotic. Got the carotenoid pigments that are being expressed there. So when I show you this plant, the same plant, if you were to look at it with only the blue energy or the green or the red or the near infrared, and notice that the healthy part of the plant in these visible bands, especially in the red and the green, is darker, because the plant is absorbing energy and it's still photosynthetically active. Over on the other side, it's less photosynthetically active, not as absorbing, it's reflecting more light. The near infrared energy is shown on the far right here, and you see some dead leaves on the table. Notice the dead leaves look a lot like the live leaves on top. What I'm telling you there is near infrared alone is not going to allow you to detect some of the things you want. You need to combine a visible energy with near infrared energy to get the best results. And this is often done using an index that goes back into the early 1970s. So this is not brand new. It's been around a long time. It's called the Normalized Difference Vegetation Index. And it's computed by subtracting the pixel values in the red or the visible band from the pixel values in the near infrared. So you have near infrared minus red in this case, and then divided by near infrared plus red. So it's near infrared minus red divided by near infrared plus red to normalize it, and you end up with an NDVI. They call it an NDVI image. And now notice that the healthy parts of the plant have a high or a bright color. The bright means a high NDVI value, and the dark areas are low NDVI. Now notice that the dead leaves on the table are no longer confused with the healthy leaves on the other parts of the plant. And this is the colorized NDVI, where we've colorized anything that's got a low NDVI value is colored in violets and purples, and anything with high NDVI is green. And when we fly over a nursery, experimental plots and so on, you're getting pixels. Some of the work we've done is at a resolution of 3 millimeters. So for every 3 millimeters by 3 millimeters on the ground, we have a pixel value or an NDVI value for the field. Okay, now I'm going to take you into some pre-processing, and then we'll come into the applications and show you how the technology is applied for phenotyping. First off, when you get the images, you should not assume that you can work with the images directly. There's lots of distortions in the imagery that need to be corrected before you can use it to do the kind of research work that many of you are probably interested in doing. The other question is, how do you correct for variations in solar elevation and atmospheric conditions? As I'm going to address a couple of those questions. Here's what we call There's two mosaics here. Some people think they can just use a stitching tool to stitch the imagery together. And the image on the left is an example of what a stitching software does. In other words, you can see the roads are not connected. The area is not connected correctly. There's 388 images in this ortho that you're looking at. This one has been done correctly with software that does actual orthomosaicing. In order to do an orthomosaic, you have to build a three-dimensional model and then stitch those images together in a way that retains the correct geometric configurations on the ground. So now you can see the roads are connected right and so on. An orthomosaic has been corrected for multiple factors that I'll explain in a minute, but its geometry is correct. Now, to show you one of the issues that you'll need to deal with, it's called the bidirectional reflectance distribution function. If you just use the individual images, then the angle at which you viewed the plot or the study plot on the ground will affect the brightness values you get from the imagery. So here is a piece of PVC pipe that's 1/4 by 1/4 meter, so it's or 1/2 meter by 1/2 meter, so it's a 1/4 meter square. And it's the same plot 9 times, viewing it from 9 different viewing angles. And then I went in and collected the data within that plot for 9 times and plotted it. And you can see if I looked at the green band brightness values, if I looked at it from the south, I get this value, from the east, I get this, and so on. You can see straight down, I get this or nadir. So I get entirely different brightness values for the same plot, all flown within a minute or two of each other, depending on the viewing angle. The same with the red band, the same with the near-infrared band. Now, if I compute an NDVI, use the green band instead of the red, or the red band, it normalizes some of this out. But there's other things that need to be done in the creation of the orthomosaic, and that is by taking the average of all the viewing angles. So when we fly imagery over the plot, you're going to see how many images we collect in a minute. A lot, in order to correct for this. The other thing is when you're using wide-angle lenses, you get what's called radial displacement. And so as you move further and further away from the middle of the picture, which is called the principal point, out toward the edges, the plants look like they're falling away from the middle of the picture. They're leaning away from the picture. And that's because you're viewing them more and more from the side. That problem also has to be addressed in the imagery. So if you just use a single image, if you were looking at plots out on the edge, you'd have very different values than plots in the middle. Now here's an example of flying a multi-rotor over some soybean, just to give you an idea again of how this can work. We usually fly at about 100 feet above the ground, but when we need really high resolution imagery, we'll fly down to as low as 30 feet above the ground. Or for a scientific term, 10 meters above the ground, if you want to be more scientific here. All right, so this is about a 2-acre nursery at Iowa State University where we flew. This was corn breeding plots. We collected in that 2-acre area 640 images, each being between 8 and 10 megabytes in size. That total data set was 5.6 gigabytes of data, to give you an idea of the size. You're going to have to deal with really large file sizes. And here again, we're showing once you get the data, how do you handle all the pictures? So we've worked to develop methods that allow you to go in and click each one of these green dots is active, so you can click on it and actually pull up an image. Now I'll pull the image, I'm going to pull up one that gives you a full resolution image. This is the kind of detail we're seeing on the study plots that we're working on here at Iowa State. And here's some soybean plots also at Iowa State. And I'm showing where we've taken some sample plots we've laid out on the plots. And in the next slide, I'm going to show you how, I want to give you an idea. I'm going to explain what these X's are in a minute. Give you an idea, if you get the software that you can use, then you can extract all the pixel brightness values within the plot. And here you see the pixel brightness values being graphed here in this particular example. Now here is that field flown on July 13th. This is 2015, August the 6th, and September the 1st. One thing we learned after we looked at the data is we wish we had flown more times between the July and the August date. That was when the plant growth was really accelerating. But anyway, these are all orthomosaics. So the next slide I want to show you is how you have to get them geo-registered. So this X is what we call a ground control point. And what I'm showing is the overlaying of the plots, the same plot at different growth stages of the soybean, and the X is not moving. If you've got your data correctly geo-registered, then the X stays in place and you know that your geometry is correct and that that you can take samples from the plots and not be sampling between the rows or whatever if you're trying to do multiple overflights at the same time in your analysis. The other question is, how do you calibrate for differences in atmospheric conditions? Here you see an overcast day that's cloudy, and you may fly on a day like that. You try to avoid flying on days that have great differences in sky conditions, but sometimes you can't avoid that. The other factor that is important is the angle of the sun. If you fly at different times in the day, you're going to get different solar elevation angles that are going to affect the number of photons per unit area that's striking the ground. So we put out a calibration panel, and I won't take the time to explain this, but if you have questions, I can give you more information on that. It's not white, and there's some good reasons for that, but I can't take the time. But what you're going to do with that is use that to help you compare one date to the next. So here you're seeing we had two calibration panels out. They were set in different conditions. Notice that this calibration panel is in a more open area, and this one is laying it down in the grass. That's something you want to avoid. When you put it down in the grass actually reflects light onto the panel and changes the brightness values. So you want to stick to an area where there's it's more clear, maybe a flat area that has very little vegetation around it with your calibration panel. Because if you don't, then you see the two calibration panels had different values, even though they were flown very close to the same time. And the average for this particular on July the 13th was 139, on August 6th it was 131, and on September the 1st it was 135. After we normalized by using the calibration panel, the values were 99, 99, and 99 on the calibration panels. But that doesn't change the fact that canopy geometry is changing, and so you're going to get differences depending upon the geometry of the canopy at the time of the overflight. Here we're showing now extracting data from each of the plots where we've laid out a transect and then use buffer analysis in a geographic information system to extract the pixel values from each of the plots. In each of these buffered areas, there's over 50,000 pixels that were sampled to give you an idea of how many values are being looked. So 50,000, 50,000, 50,000 for all those plots. And then you can do, once you've got the data, you can start doing, we put the data into cluster analysis to do classification. We calculate basic statistics, means and standard deviations, and so on. And then you can start classifying the plots if you want. This is the classification of those plots using the red edge band, which is a band that is in the area between the red and near infrared region of the spectrum. And this one's using a green band and showing the differences. Here we've computed the standard deviation of the red edge, and here the standard deviation of the green, and showing the differences in the plots. And the agronomists or the plant breeders are now putting these kinds of data into their algorithms to see how it allows them to discriminate differences in genetic types that are creating different phenotypes in their plots. Here's an example of doing some work on sorghum. By the way, this is an orthomosaic. It's not a single picture. You can see how the spliced edges are all jagged. That's because we've got hundreds of images going together to make the orthomosaic. This was the multi-rota that was flown. And to give you an idea of the detail, you're looking at the paper sacks now that are on the different plants to help cover up the pollen or the seed, excuse me, the pollen heads and so on. Now, one of the things we did here is we wanted to separate. One of the questions was, are we increasing on the genetics, are we increasing the phenotype, are we selecting for phenotypes that have the top leaves of the plant more erect and the bottom leaves more decumbent or horizontal? That way, if you've got them more erect at the top, as probably most of you know, you're going to get increased sunlight deeper into the canopy, which hopefully will increase biomass production. This was a biofuel study, so they were trying to select for genetic types that increased biomass. So we classified the plots into areas. The green areas are where we have upper leaves, the yellow is the middle part of the canopy, and the purple is the lower part of the canopy. The white areas are where we've masked out the bare ground to keep bare ground from being a factor in the analysis. I guess just to zoom in of what I just showed you. And here we took a tract of 62 plots, and we then put all those plots out and looked at them and extracted the statistics. So this graph here is showing for each of those 62 plots the percent cover by the lower leaves, and the green is the upper leaves. We've left out the middle leaves in this example to make this graph a little less complex to look at. But what you're looking at is the variation in the genetics that's creating different phenotypes across those different plots based upon the percentage of upper and lower leaf visibility. So this is an example of just 4 of the plots we pulled out to show you. What this is showing is the classification by the level in the canopy, and this is the color infrared image. The way you look at this is the redder it is, the greener it is, and the darker are the shadows. Now, canopy geometry is going to change the shadowing components within that canopy, which then means that that shadow can become information to you as well. But what we've got is the plot up above is showing that in plot 24, it had a higher percentage of cover that was visible from the drone in the lower canopy and less that was visible from the upper canopy. This plot 41 was also the same, but plots 27 and 47 were the opposite. Less lower canopy visible in these two and more upper canopy. So if I were genetically selecting, if I wanted to select for the plots that have more vertical leaf structure at the top, I would select for plot 24 and 41, and I would probably exclude 27 and 47. Now, another measurement that's used, this is actually a measurement I used to teach an area called landscape ecology. And so we would use a metric that's used in landscape ecology to determine how connected the landscape is for wildlife to move from one area to a next. So using a metric called contagion, we then looked at the amount of leaf area that was in the upper canopy, or that is contagious, meaning it's connected, compared to the amount that is less contagious or most contagious. So what you're seeing in these plots is the different amounts of contagion. So if we go to this next plot, which see again, if we plot the contagion, you can see that the upper canopy is in the green here, this green. There's not as much in this upper canopy as there is over in plot 27 and 30, which means that these have more upper leaf canopy visible, and these two plots, 23 and 24, have less upper leaf canopy visible. And if you're interested in counting seed heads, the blue, we've classified the blue, and that's basically the seed heads on this sorghum, on these sorghum plots. Okay, another factor you may be interested in is texture, because texture becomes an indication of canopy. The canopy, the percent cover, the density of the plants, the canopy geometry, whether or not you've got a very uniform canopy or whether it's very ununiform. So we can just lay a transect across. In this case, I've taken about a 1 square foot area from winter wheat and a 1 square foot area from canola. And and then I've laid a transect across them, and the software then builds a graph. And the next slide is showing a zoom in on the profile for the different bands, blue band, the green band, and the near infrared band. And you can see on the winter wheat, you can see canopy and then shadow, canopy and then shadow. So what you're seeing is the high peaks on here are where you've got leaf area that's visible to sunlight, and the dips in here is where you've got a shadow component in the canopy. So canopy, shadow, and so on. So you're seeing that differences in texture. You're also seeing that it's very similar in its pattern for all three wavelengths that were being looked at. The canola, on the other hand, has a different type of geometry that makes it different from the winter wheat. So this kind of a measurement could be used on different plots of winter wheat or whatever or corn or whatever you're breeding to look at the differences in the geometry of the canopy. Now, another question is biomass, and often biomass can be, not always, can be correlated with yields if you're looking at grain yields and so on. I don't have the geneticists that are looking at that now with some of the newer data that we've flown, but for last year. But here's the calibration panel, and this is grasslands in Kansas, and I want to show you how we've looked at the correlation. Biomass is often highly correlated with the vegetation index, or the NDVI. So this is flown with the drone at 5 meters above the ground. This is at 50 meters above the ground. So you see the PVC pipe here and here in the calibration panels, and these are my students and I out collecting field data. We went into those quadrats then and clipped the plots to get a biomass estimate, and then build a regression model. Obviously it's a curvilinear function. And we plotted the NDVI against biomass and got an R-squared of 0.9. So that R square is going to vary depending a lot upon plant types and a lot of factors. But I wanted to show you this, that it can be strongly linked if biomass is an issue of interest to the people who are listening here. One of the other factors that, this one was actually flown with the Cessna. This particular genetic breeder has about 33,000 plots. I asked him, how do you get out to look at 33,000 plots? And he said it's very difficult. Often they have to go to the ones they think are most likely their best indicators. But what you're seeing here is differences in the redness of this field, these plots. This is corn in Iowa. And the brown areas are places that are already, the corn has already gone into senescence and is browned down. The red areas, the corn is still photosynthetically active. So what the geneticist used this for is to go out to those plots where he saw that they had browned down earlier, and he wanted to make sure he got yield data from those plots to see if he was getting still high quality yield as well as high quality grain on a plant genetic type that was genetically advanced and when it matured. And so that's how this kind of data were being used. And you can see differences in planting times and so on up in here. Okay, another thing is that when you do lots of overlap between your images, the software that's out there now uses a technique called structure from motion. And structure from motion is different than LiDAR. Maybe some of you are familiar with LiDAR, which is a laser beam that beams light to the ground and then determines how long it takes for the beam to go from the transmitter to the ground and back to the receiver, obviously moving at the speed of light. But that's the way they do elevation models and calculate the height of trees and so on, is with LiDAR. A newer method that's out there that works very well if you've got enough gaps in your canopy is called structure from motion, and it uses the overlapping images to triangulate from the same point. So for example, when we fly that high density imagery that you saw, we may have 15 or 20 images that have the same exact spot in the field in all 15 or 20 images. So the software then goes in and calculates the angles and uses the solid geometry techniques to determine the distance and build a three-dimensional model of the surface. And in this case, these models, if you collect good ground data in terms of the actual elevation points on the ground and get good surface data, then you can subtract what's called the digital terrain model from the digital surface model and calculate the height of your plants. And in this case, we've run a profile across the sorghum field, and the geneticists went out and looked at the variation that we were picking up along that transect and said that it looked very promising to them. They were very excited about it. The pixel size on this overflight was 7 millimeters, and with correct calibration, you can be usually within plus or minus 1 pixel of accuracy on your elevation data. All right, some of you may be interested in thermal. I thought I would throw in just a couple of slides as I wind up here on thermal, and that is that this is a cornfield, this is a thermal image, and this is an NDVI image. And you can see that the two look very similar to one another. This is a cornfield in Nebraska. There were 12 thermal images flown over a field near this one, and this was flown on May the 7th, and you can see the different dates here going across, and you can see that the thermal imagery is highly variable from date to date. And some people may wonder what's going on here, and what you're seeing here is wind speed, or the velocity of wind going across the field. So using thermal data can be a challenge, especially in an area that's windy. If this field, a shadow from a cloud went over before the thermal imagery was flown, and the shadow changed the temperature of the canopy enough that it masked out the plants, and you're seeing the shadow effect. So, and here you're seeing lots of wind velocity in here. So thermal imagery can be very useful, but it also can be a real challenge to use. All right, that's all. I think I probably, well, it looks like I left some time for questions. So I've covered it pretty quickly, but I'd be glad to take any questions.
Okay, great. Ryan, so we've got one so far. So yeah, I want to encourage folks, yeah, any questions you have, feel free to put those in question and answer, and we can chat about those. So Ryan says, what size of alley is needed between plots to get accurate structure for motion? And then in parentheses he says cotton images.
Oh, for which kind? For cotton?
Yeah.
Oh, that's a real good question, and I don't have a good answer. I will tell you that on the work we were doing, the alleys were obviously just left as they normally are put in on the sorghum and the corn. But we have one of our dealers is working on cotton, but we're not doing structure for motion on cotton yet. That is an area that's needed because obviously you want growth retardants on that cotton and figure out where you want to put it for doing precision agriculture types of applications of growth inhibitors. I wish I had a good answer. I think that I would start off just using the normal alley sizes and see how it works, and if it doesn't work, then try it a different way. Or if you want, I mean, if you're obviously not going to get a farmer to change the alley sizes very easily, but for your research you probably could. You might experiment with that some.
Sarah says, have you found any techniques to remotely sense ear height in corn?
The ear of the corn, we haven't tried that. It would be a challenge. Basically, if the ears are covered by the corn leaves or blades, then you're not going to see the ears. You're not going to catch them. So I'm not optimistic that that's going to work very well, because quite often those corn ears are going to be covered up by higher level canopy.
Okay. Josh says, have you had any chance to apply this to specialty crops like orchard trees?
Orchard trees to get the height of the trees? Is that what you're wondering?
Well, he just it was a general. Josh, if you want to add any context there, please do.
Are you wanting to know about the NDVI on orchard trees?
Yeah. He didn't specify that. So, Josh, if you want to add some more context, feel free to do that.
Let me just assume he's talking about detecting differences in the plants in terms of their canopy. Anything that changes the pigmentation of the plant, or in other words, the chlorophyll, should be detectable by the remotely sensed measurements. So if the plant leaves are going chlorotic, and anything that stresses the plants are going to cause that plant to usually go chlorotic, whether it's water or nutrients or disease or whatever. So what you want to do is use indices that are sensitive to those pigmentation changes. As that plant goes yellow, it's changing from chlorophyll to carotenoid pigmentation. And there are different vegetation indices that help pick that up. Now, if you're wanting to do structure for motion on the geometry of the canopy, you'd be obviously doing mostly the surface geometry of the trees. I'm not sure how much you'd be doing any inner canopy geometry, but it should work fine on trees.
Yeah, and he followed up there and said anything tree height, and said more importantly, chlorosis. I may be saying that wrong, or tree volume. So
chlorosis, yeah. And I think I answered that question.
Okay.
If I didn't hit it right for you, shoot another question. I'll be glad to try to address.
Okay. She says, what's the resolution you can achieve with the current drone technology?
With the multi-rotor, where you can fly low and slow, you can get down, like I said, we've flown 3 millimeter resolution. If you're flying at 400 feet, you're going to get about 1 inch or 2 centimeter resolution. One thing I will tell you is that there's a lot of cameras out there, and there's a lot of bad cameras out there. You need to be really careful, and I just didn't take the time or think I would have the time to go into that, and certainly did not want to start picking on any vendors out that are selling cameras. But I will tell you that that you want to get a high quality single lens reflex camera as opposed to a point and shoot if you're doing this kind of work. And you can fly that kind of a camera on a multi-rotor, and you can fly it low and slow, so you can keep from having what's called motion blur. So that would be my suggestion. Okay. And that you get a camera. I'm probably going to get someone who's going to beat me up for this, but for 25 years I did research in hyperspectral. I found when I do partial least squares regression analysis and principal components analysis on the bands, when I look at 1,000 to 2,000 bands, I find that it all boils down to 3 portions of the spectrum on vegetation. Usually a green, a red, and a near-infrared band are the optimal regions, and if you've got a camera that has those, you're going to explain about 95 to 99% of the variation in your data. So I would be scared to death to put a $40,000 hyperspectral scanner on a drone. I speak all over the world on this topic. I've spoken to about 20,000 people in the last couple of years. And when I ask how many have flown a drone, maybe 5–10% of the hands go up in the room. The next question I ask is, how many of you have crashed your drone? And all the same hands go back up. And then I ask, how many of you picked your drone up in a black garbage bag? And all the hands go back up again. So if you want to fly a $40,000 scanner on a drone, you're a lot braver than I am. Okay.
Okay. Ryan asked, get that again. What technique was used to distinguish sorghum heads from leaves? Was it color differences? And then I guess he says, is this possible for cotton?
Looking at cotton bolls is what they're wanting to do.
Man, he didn't say cotton boll. He just says, is it possible for cotton?
Well, I can tell you that I'm working on some work with Texas A&M, and they have been counting cotton bolls with drone imagery, but we're up in the corn and soybean region, so we haven't been doing the cotton bolls, but I know that Texas A&M has. The detecting the seed heads on sorghum, it was just a basic classification approach that we used. The sorghum seed heads on the ones that we looked at were spectrally different from the leaves enough that we could tell them apart. I don't remember which bands, but again, we only had 3 bands, and we probably used all 3 of the bands in a multispectral analysis approach.
Okay. Let's see. Mart says, any studies on scouting for insects using drone technology?
Yes. I didn't show any here because I was talking about phenotyping, but we have. And we haven't done the kind of control studies that I'd want to do as a scientist would do. But I can tell you that anything, again, insects are going to be eating the chlorophyll. So it's going to lower the chlorophyll density, and so you're going to pick it up with the imagery. Again, all you have to do to answer most of your questions is say, does it change chlorophyll density? If the answer is yes, or does it change the pigmentation? If the answer is yes, then whether you can detect it or not, most of the time my answer will be yes. The one thing we found interesting, we flew alfalfa in Arizona. We flew 1,000 acres, and we landed the drone multiple times while flying the alfalfa. And one of the last times we landed in the lushest part of the entire 1,000 acres. We picked it up. It was the drone was completely covered in green bugs or aphids. So one thing that we learned from that is that the aphids are like we are. They want the beefsteak. They aren't going for the beat up stuff. So if we assume that the bugs are in the bad parts of the field, we may be wrong. They may be actually in the best parts of the field working on the good stuff. So we found them there in the very best parts of the field.
Okay, great. Let's see. Linda says, are there available tools to overlay geographic plot data, i.e. nurseries planted by GPS with large image data, or do you have to manually draw polygons for each plot?
No, we just did a project where we laid 15,000 shape files onto a plot, and we've written software that allows us to extract the statistics from those. So if we have shape files, we can drape those onto the imagery, but you obviously have to have, if you're doing multiple dates, you have to have high precision in your geometric, in your register. It's called georegistration. If you're just registering image to image or mosaic to mosaic, we call it georegistration. If you're actually trying to tie it to ground geographic coordinate systems like lat long or whatever, then it's called georectification. You don't need to do georectification usually unless you are trying to tie coordinates from your GPS that are in a coordinate system. Then you do need to georectify the images to the ground, and then you have to georegister date to date, and then you can overlay your shape files onto it and extract the values.
Okay, great. Andrew says, regarding camera and payload requirements of multi-rotor drones, are professional series, e.g. DJI S800 drones required as opposed to consumer drone solutions, e.g. DJI Phantom?
Okay. The Phantom is going to collect some great natural color imagery. The Phantom has a very wide angle lens, so you're going to have geometric distortion in your outer edges of your picture a lot more. It works beautiful for doing three-dimensional models. I am less optimistic about how well it would be for doing high throughput phenotyping, where you're extracting spectral values. There are vegetation indices that are created for natural color, and some of them are pretty good. But the DJI 800 we showed there actually is an older model. We now use the 900, and the reason we use that is we can fly a converted camera, and I didn't talk about that, but you can have your cameras converted so they are natural, so they can capture color infrared imagery. But in the process of converting these cameras, you want to make sure you're working with a camera with a bigger detector, is our views on that. And the reason for that is that the bigger the detector, the more surface area on each of the individual detectors, and you're going to get a stronger or higher signal to noise ratio that allows you to detect subtler differences in the vegetation reflectance. If you get a small camera like this on the Phantoms and so on, they're using a really small detector. They're great for video. They're not bad for doing the three-dimensional modeling, but they're not, in my experience, not what I would want for doing high throughput phenotyping.
Okay. All righty. Victor just sort of has a broad or general question. What do you see as the biggest limiting factor? What do you see as the biggest limiting factor to seeing more widespread use of drones in agriculture?
Oh, well, there's a couple of them. One of them is that the people who fly have got to get FAA certified to do it legally, and your universities will will be coming down on you pretty hard if you aren't, because you're making them legally liable for a lot of potential lawsuits. You want to make sure you have good insurance covering you. It's not uncommon to have a drone get away and fly off into the next county and land somewhere. Can be a challenge there. The biggest factor is going to be probably getting people up to speed on what to do with all the data once they've collected, and make sure that they're collecting it in a way that is of scientific quality, not just pretty pictures. It's real easy to click. Well, it's not even real easy. A lot of people are collecting pretty pictures, and then when they get through collecting it, they find out that they didn't collect it in a way that allows them to do the scientific research they want. So knowing something about remote sensing is pretty critical too. If you don't have the remote sensing background, there's a lot of ways you can do things with the data that violate some of those problems I talked about. Not being aware of the geometry issues, the bidirectional reflectance distribution function, the radial displacement, and so on, that can plague your data. So those are issues. Yeah, there's a lot of challenges. I don't want to sugarcoat this. It's a very exciting area. It's certainly the way it's going to go. The National Research Council said that the biggest bottleneck to genetic breeding is the high throughput phenotyping, and we've got to get a handle on the high throughput phenotyping, or we're going to be slower at being able to move our genetic varieties into the market. So I think this is going to be something that's going to take off. We're pretty early in it. I started doing this 4 years ago, and working with Jesse Poland at Kansas State University. I'm working with Jan Ming Yu at Iowa State and several others here. But there's a lot of exciting opportunities. Make sure you ask a lot of questions. Again, you've got my email address. That's my cell phone number. Literally, if you call that number, it's going to ring my phone right in front of me, so I'll pick it up.
Okay, we've got a few more minutes. Let's see if we can squeeze a few more questions, folks. Feel free to ask. Feel free to ask questions. If we don't get to them, they'll be recorded in our reporting, and we can work with Chris and Kevin to maybe get you some answers via email. Josh says, can you explain why? Can you explain why structure for motion is better compared to LiDAR again?
Well, structure for motion is a lot easier to collect the data. It's a lot cheaper. For example, you can do structure for motion from that Phantom, which is about $1,000 multi-rotor. You can do structure for motion and do some beautiful work with that. When I said that I'd be less enthusiastic about using that, it was in trying to do the spectral analysis. When you're looking at the 3D stuff, the Phantom should work great. But the structure for motion is different from LiDAR in that LiDAR is an active sensor. It sends a beam of light like a laser pointer. It's just like when you turn on a laser pointer. It's sending a beam of light out, striking the ground, and coming back. So when you have dark spots or shadows in the canopy, the light is more likely to penetrate down into the canopy and hit the ground. And you want the ground, you want pixels that hit the ground as well as pixels that hit the canopy, because then you can figure out what the ground elevation is as opposed to the canopy elevation, and subtract one from the other to build your elevation or your height of your plants. If you're using structure for motion, if you've got a very dense canopy, you're only going to get the outer surface of the canopy, and you're going to have to assume that the places in between the canopy are representative of the elevation of where the plant is. Now, if you fly imagery before the plants start growing and get a surface model of your field, then you can use that later on, and you don't have to worry about that. Structure for motion will work fine. Structure for motion is a lot easier to get computed. You don't have to buy $1 million system. The systems are being flown on $1 million planes. Now, I know there's some drones that are starting to come out with laser capability, LiDAR capability. I haven't had a chance to evaluate that. Don't know how good it is yet. But you need lots of points. Even LiDAR has a problem getting through the canopy.
Okay. All righty. Just for folks, we're going to take another question or two, and then we'll wrap it up. I put a link to the Learn event in at extension.org inside chat. So again, the recording will be available there within a few days. James says, was that yellow ground target you used? What was that, a yellow ground target you used, and why not white?
Yeah, well, that's a good question. White will cause the sensors on your camera to peak out or saturate. So if you have a spectral on panel like we normally use for hyperspectral analysis, the white panels will cause all the brightness values over the panel to just peak out. And so if you calibrate the camera so that it doesn't peak out, then all of the imagery over the plants will be so dark you can't see them. So what we had to do is experiment with different colors of paint to get the right paint that gave it so that none of the values, the blue, green, and red, or the near infrared, didn't peak out. The color that you're seeing there is called Italian olive. If you want to write it down, you can get it at Home Depot. It's a bare paint. You want flat. You don't want gloss or semi-gloss. You want flat. And it's Italian olive. But you can do this by just going and getting paint chips from the paint store and laying a whole bunch of them on the ground and shooting pictures with your camera and seeing which ones cause it to peak out. But the Italian olive was kind of a middle ground that didn't cause saturation. Okay.
All right, I think we'll have this be our final question. Nolan says, do you have suggestions regarding software for determining structure from motion? What about for measuring the models generated?
You kind of went, the sound went really crazy a couple of times. I didn't catch all that question. I'm sorry. Could you repeat it?
Sorry about that. Can you hear me okay now?
Yeah. Uh-huh.
Okay. It says, do you have suggestions regarding software for determining structure from motion? What about for measuring the models generated?
Okay. The software, there's two major pieces of software out there that are available. The two that you can use is one's by Agisoft. And students, you can get experimental versions for pretty cheap. It runs about $300, maybe $500, I can't remember, for that software. And you can do a lot of good work with Agisoft. You can get a loaner and play with it for a while and see how it's going to work. You can also get Pix4D. It's about $9,000. It's very expensive. And Pix4D will do the three-dimensional modeling you're wanting to do. If you want to extract the data, a lot of that right now is pretty customized. It's you can use GIS software programs like ArcGIS and so on. It's cumbersome and slow.
All righty, great. Okay, thanks everyone. I think that's all the time we have for today. Again, I put a link in chat. Chris, any closing words on anything?
Nope, I think you all covered it well. Thank you again for being here.
Okay. All righty, Kevin, thank you so much for your time.
Thank you for your time. And there's my email address. If you didn't get me to answer a question, pop it to me. I'll answer it.
All right, thanks everyone. Bye-bye.